All right, everyone, thank you for joining us for the Human Virome Technical Webinar. We appreciate all you taking time to meet with us. If you need more information on the program, you can find more at our website at commonfund.nih.gov/humanvirome. And you can also email us any of your questions at humanvirome@od.nih.gov. And as we get started, I just want us to go over a few housekeeping items. This webinar is being recorded. Just be aware of that. And the format for this webinar will be, we will start with some brief presentations on the program and the different initiatives. And then we will follow that by a Q&A session. We ask that you please submit your questions into the Q&A box, the WebEx Q&A box. That way we can mark off questions that we've answered, as well as assign them to specific people. So please use that box and use the chat for technical issues that you might be encountering. WebEx can be a bit finicky sometimes, especially with audio. So if you start having audio issues, usually if you log out and then log back in, it will resolve those. We also ask that you please stick to questions that are going to be of broad interest and utility for the group. So if you have very specific and very narrowly focused scientific or situationally specific questions, we ask that you not bring it up here, but please email us and we'd be happy to talk with you offline. For additional questions, you can email us at humanvirome@od.nih.gov. And this webinar is being recorded and we will post the recording and the slides that we're using today on our website at commonfund.nih.gov/humanvirome. And see our website for more information and guidance, including an FAQ page. All right, so want to introduce our panelists for today for this webinar. So from the Common Fund, I'm Becky Miller. I'm the program leader for this particular program. We also have Sonika Nagoso, who has joined us from our office. And David Bullweg is our budget person. And I don't think he's here today, though. For the characterization centers, we have Dr. Roberto Flores Humia from the National Institute on Aging and Catherine Kim. We also have, hopefully she'll be joining us later, Stacy Carrington Lawrence, also from the National Institute on Aging. For tools and methods development, we have Dr. Amanda Milillo from NIDCR. And for the functional characterization initiative, we have Dr. Haesuk Kim from NCCIH. And here to talk about the coordinating center is Dr. Emmanuel Mongodin from NHLBI and possibly Shimian Su, also from NHLBI. You're welcome to type in your questions if you have specific ones for the initiatives or for a specific person. You're welcome to type in their names and we can assign those to them. I want to give you a brief introduction, though, on the Common Fund. So the Common Fund is the one who has put together this program on the Human Virome. And part of the mission for the Common Fund was that it was really created to help push forward the broad NIH mission and to help accelerate research and overcome barriers that may be acting as roadblocks to scientific discoveries. So really the Common Fund was meant to really catalyze discovery across the broad mission of the NIH. We advance areas of relevance to the NIH, but also that are of relevance to multiple NIH institutes and centers. And the goal is ultimately to really spur advances that would not otherwise be possible without this initial strategic investment. So we fully understand that we're not going to be able to solve all the problems. So Human Virome, we understand we're not going to be able to categorize everything, but we want to help really give this area a boost and help push things forward than it would be possible without such investment. So Common Fund programs have some different areas that they tend to focus on, but they all make substantial investments in a time-limited, goal-driven program. So all Common Fund programs are mandated by Congress to be no longer than 10 years. They have to have discrete goals with milestones and deliverables at the end of the programs. but they're all meant to really change the trajectory of an area of science. So they can do things like accelerate emerging science, remove roadblocks, enhance a workforce, or support high-risk, high-reward science. And we identified the human virome as a prime area that could really use that common fund investment. So we know that the human virome is very large and diverse with greater than 10 to the 13 particles per person. And the commensal virome is largely understudied, and it's not known about its interactions with the human body, as well as its impact on long-term health, human health and disease. So as you can see from the schematic here, this shows different anatomical sites and some represented viruses that have been identified there. And so we want to really expand on this and see if we can characterize more of the human virome, as well as identifying some of the impacts that things like our diet, medicine that we're taking, our geography, our age, how that can impact the human virome. And so this program ultimately has the goal to characterize the human virome and define its role in health and disease. And this will be done through 4 initiatives. The first is to characterize the human virome in longitudinal demographically diverse cohorts across the lifespan. We will also be developing tools, models, and methods that will allow us to interrogate and annotate the human virome. We will also study interactions between the virome and the human host, as well as with other components of the human microbiome. And we have to create a coordinating center to help manage all of this and to really make this all possible. So we need a coordination center that will serve as an organizational hub. We also want to create a data portal and a human virome reference data set, which will be part of the purview for this coordination center. And an important thing to note for this program and all of the initiatives that and funding opportunities that have been released for this program is that they require consortium building. So like everything is focused on building this consortium. So please note that as you read the funding opportunities, it talks a lot about how we have this extensive collaboration is required. We're using cooperative agreements, which means that NIH will be substantially involved with the work. And our consortium will be overseen by a steering committee that's going to be made up of the investigators and NIH staff. We also require that these investigators attend the meetings and participate in the meetings for the program. And we are aiming to have a kickoff meeting about 3 months after the awards are made. Anyone who is a part of this program is also expected to abide by consortium developed policies and guidelines, which can include policies on the standards for collecting or analyzing the biospecimens, as well as the resources, different methods and data collection, all those types of things. All of the data that's developed or resources and tools, they're all expected to be deposited in the data portal and shared within the consortium. And you'll read in the funding opportunities for all of them except the coordinating center that we do require collaboration projects. So this is between investigators from different projects, but within the consortium. And that's really, once again, to help build this consortium environment. And please note that we also require a plan for enhancing diverse perspectives in the applications. So from there, I will go ahead and hand this off to Roberto to talk more about the characterization centers.
Thank you, Vicky. Well, good afternoon. My name is Roberto Flores, and I'm the Program Officer for RFA 2319, the Human Virome Characterization Centers. This RFA is a U54 mechanism, and clinical trials are not allowed. Next slide. The goal of this RFA is to define the human virome and its dynamics, utilizing longitudinal diverse human cohorts across the lifespan by sequencing the viruses that comprise the human virome and providing an accurate estimate of its richness and complexity. Next slide. The Virome Characterization Center's responsibilities are to identify both eukaryotic and prokaryotic viral composition within diverse human populations across age, ethnicity, sex, gender, and geographic location. And this viral characterization should be done in people without evidence of overt disease. The VCCs should also efficiently study the human virome across tissues by planning for the collection and use of biospecimens from at least 2 anatomical priority sites. These priority sites are blood, respiratory tract, gastrointestinal and urogenital tracts, including urine and stool, oral cavity, central nervous system, sampling the cerebrospinal fluid, and skin, including hair follicles. Tears and breast milk are acceptable biospecimens, but these would be in addition to any of the 2 priority sites. Next slide. Additional responsibilities is to collect, process, and analyze the virome and human fractions of each sample and determine the relationships between the virome and other components of the host. Categorize virus taxonomy, eukaryotic and/or prokaryotic and bacteriophage, and characterize newly identified viruses, and determine factors that influence virome composition and its host dynamics. Utilize an adequate number of samples to ensure statistical power for hypothesis testing. Next slide. This notice of funding opportunity uses the multi-component U54 mechanism, and each virome characterization center will include 5 cores with well-defined and distinct function and working synergistically. And the cores are listed here. It will be an administrative core, biospecimen collection core, biospecimen analysis core, data analysis and submission core, the ethical, legal, and social implications, or ELSI core. These 5 different cores should be integrated to achieve the overall goal of the virome characterization center. Next slide. So the administrative core is led by the contact program director or principal investigator of the virus characterization center. The main responsibility is to provide leadership and decision-making activities of the center by developing standard operating practices, developing metrics for data generation and metadata standards, participate in cross-site studies, and engage in cross-training and guide development of data analysis and visualization tools. Additional responsibilities include the coordination of various components of the VCC, coordination with other VCCs, and facilitating communication and coordination with the Human Virome Program Data Collaboration Center. Next slide. The Biospecimen Collection Core has the objective to collect and process samples from existing cohorts for the isolation and characterization of the human virome as established by the Human Virome Program Consortium and the steering committee of the Data Collaboration Center. The biospecimens collected must comply with the following: having sufficient quality along with extensive metadata from individuals with diversity in gender, race, socioeconomic status, geography, and other relevant diversity parameters, represent the human lifespan with different age ranges as indicated, birth to 5, 6 to 10, 12, etc. It is not expected that every single VCC should assess all age ranges, but the consortium as a whole is striving to be inclusive of the human lifespan. Should also have an absence of overt disease or to be of a certain health status. And preference will be given to appropriate specimens from longitudinal cohort samples and phenotypic data. Additionally, the BCC should develop specific quantitative annual milestones for collecting, processing, annotating, preserving, and classifying biospecimens. Next slide. The objectives of the Biospecimen Analysis Core is to use human biospecimens to identify and provide in-depth characterization of both eukaryotic and prokaryotic viruses, replicating and integrated viral particles, proviruses, and viral proteins, and to characterize viruses that will contribute to building the human virome database. The responsibilities are to generate high-quality data with resolution, content, and throughput to comprehensively discover viruses contained within human tissues and biospecimens, establishing and optimizing SOPs for the collection of relevant metadata, including assay validation and benchmarking and metrics for data quality control, and work closely with the Data Analysis and Submission Core and Data Collaboration Center. Next slide. The objective of the Data Analysis and Submission Core is to construct the human virome database produced by the VCC and making it available through publicly available data repositories. The main responsibilities of this core are data annotation, curation, analysis, and submission to public repositories, building data sets for public repositories with the characteristics listed here, and use of software and analysis methods, including advanced AI methods, to facilitate data analysis and enhance scientific rigor, reproducibility, and usability. To develop and implement consortium-wide open data and metadata with standards, quality metrics, data elements, imaging and omics data, and annotation. Cross-validation of assays within and across VCCs and interpretations of data generated. The design and conduct of at least one preliminary study for functional characterization of identified viruses. Slide. So the objective of the Ethical, Legal, and Social Implications, or ELSI core, is to protect participants, investigators, and NIH staff in matters of privacy, safety, and legality by providing guidance on ELSI and policy issues. The ELSI core will work closely with the Biospecimen Analysis core and the Data Analysis and Submission core on key issues. Filtering human contamination, for instance, uniquely identifiable genomic sequences from donors, keeping up to date on technological advances that will make it possible for publicly available data to be traced back to enrolled participants, perform research on potential risk to people, including the potential uses of data and the application of the Genetic Information Non-Discrimination Act, GINA, to the Human Virome Program data, and assist the data analysis core in the return of results to participants. Next slide. These are some budget considerations. The NIH Common Fund intends to commit approximately $20 million in fiscal year 2024 and $20 million in fiscal years 2025 through fiscal year 2028 for this RFA. The wording intends to commit refers to total cost, not direct cost. It is anticipated to have 4 to 5 awards. And investigators for each VCC should set aside $250,000 in total funding in project period years 3, 4, and 5 to support multiple collaborative pilot projects, each of $50,000 per year for up to 2 years. The budget should include funds to support travel for PDs and PIs and pertinent members of the center to attend annual consortium meetings, consortium activities, and workshops. Be aware that NIH may modify budgets on award. Next slide. In terms of eligibility, foreign institutions are not eligible to apply. Non-domestic, non-US components of US organizations are not eligible to apply, but foreign components are allowed. For-profit organizations and NIH intramural program are eligible to apply, and institutions may apply to one or more RFAs of this human virome program. Next slide. So some administrative details. Be aware there will be substantial NIH programmatic involvement in individual projects and consortium activities. The Human Virome Program RFAs are one-off announcements with no revisions or appeals, and no late submissions are allowed. Reviews will be in special emphasis panels. Please pay attention to the review criteria given in the RFAs. And there is a link to frequent FAQs that is available in the Common Fund website, and the link is provided. Next slide. So these are important dates to consider. LOI due date is October 17. The early submission date is also October 17. Applications are due on November 17, 2023. Peer review will be done in March of 2024. Advisory Council will be in May of 2024, and the early start date of this program is July 2024. Thank you.
All right, we'll move on to Amanda.
Thank you. Thanks, Becky. So this next section will be on RFA RM 23018, which is the second initiative of the Human Virome Program on developing novel and innovative tools to interrogate and annotate the human virome. This is also a UO1, or similar to the next section. This is a UO1 mechanism, which clinical trials are not allowed. Next slide, please. So, as I already mentioned, the purpose for Initiative 2 is to address the technological challenges that are currently hindering robust integration of the constituents and functionality of the human virome. So this NOFO is specifically focused in two areas. One is the development of innovative and novel tools, methods, and models to overcome major challenges in identifying and characterizing human viruses. And two, the development of computational biology and bioinformatic tools to enhance the analysis and visualization of the human virome. Next slide, please. So specific areas of research that this NOFO is interested in are listed here and on the next slide, but these are just examples and are not limiting to only these areas. So to go through some of these, this would include the tools or methods for viral isolation and detection, tools and or methods for viral quantification, techniques for in vitro viral propagation, replication, infectivity, and or visualization, culture systems with high throughput capacity, enrichment techniques for downstream sequencing, laboratory or in silico approaches for eliminating environmental background and contaminating sequences, approaches for human and animal procurement and storage. Next slide. And to continue this, in vitro assays and animal models for virome dynamics, methods and tools for host range prediction of sequence data, viral interaction detection, ecology-based analysis of the host viral population dynamics, in silico methods, for example, computational, statistical, power analysis, et cetera, and functional annotation of uncharacterized regions of viral genomes, in silico methods to compare virome across studies, and methods to identify sequencing errors leading to false identification of viruses or variants and lack of matches in the current viral databases. Next slide. So as part of the research plan section of the application, which is section 4, we ask that you describe how the tools, the technologies, or methods that you are planning to develop and use will overcome major challenges in identifying and characterizing viruses. So this should be clearly laid out in the application. In addition, in situations where improvement on existing tools, technologies, or methods is being proposed, applicants should describe how those proposed new tools will be significantly faster, cheaper, more sensitive, or robust than the existing methods, tools, technologies. A description of how the PIs plan to work collaboratively with the other components of the Human Virome Program Consortium should also be addressed in this section. Furthermore, project milestones are goals that are create decision points in the project should be clearly indicated as criteria for success. Yearly milestones should provide clear indicators of the project's continued success or emergent difficulties, and may be used to evaluate the application not only in peer review, but also in consideration of awarded projects for funding of non-competing award years. Next slide. So the key dates for this RFA are the due date is November 14th. Scientific review will occur in March of 2024. It will go to the May Advisory Council in 2024 with the earliest start date of July 2024. And no late applications will be accepted under this notice of funding opportunity. Next slide. As for the budget information, the NIH intends to commit approximately $4 million in FY 2024 for this NOFO. So approximately 8 awards are anticipated. Of course, this is contingent upon availability of funds and receipt of sufficient number of meritorious applications. And the award budget for each application is limited to $350,000 each year in direct costs. And the project period should really be determined by the scope of what the PI is proposing, with a maximum of 4 years. So just to also remind folks, as we heard in the last talk, that you also want to look at Section 5 of the RFA very closely for the review criteria and make sure that all of those points are addressed. Thank you.
Okay, Isaac.
Okay, hi everyone. My name is Hasu Kim. I'm also lead of this RFA RM 23017, title on Human Virome Program, Characterization of Functional Interactions between Viruses and Human and Microbial Hosts, which will utilize the U1 mechanism. A clinical trial not allowed. Next slide. As mentioned in the title, the goal of this RFA is to study functional characterization of human virome interactions within human and microbial hosts. Within this RFA, there are 2 specific research areas that has to be focused in the application. The first is to characterize tissue trophism and host reservoirs for all resident viruses. The secondly, the application should define and characterize the functional interactions of the resident viruses within human host tissues and the microbiome. So regarding what types of the viruses or virus will be considered as part of human virome is all commensal viruses that are commonly found in or on humans are acceptable if studies demonstrate or also expand our knowledge that will be gained on how virome interactions within human and microbial hosts impact human health. Next slide. These are the few examples of the application topics regarding tissue trophism and host reservoir, which include identification and characterization of host cell and tissue receptors that are recognized for viral engagement, other cell entry factors that are needed for establishment of infection and persistence of human virome, and lastly, host factors that are important for virus life cycle maintenance and or reactivation. Next slide. For the application on virome functional interaction within human and microbial hosts, those examples are listed here. The first one is the virome and immunity. The second, host virome cellular and molecular interaction, followed by bacteriophage and microbiome interactions impacting the human host. Viral dynamics, evolution, emergence, or co-infection will be examples of these types of the topics of interest. comparative studies of viral homologs, and lastly, uncharacterized RNA virus interactions within a human and microbial hosts will be the examples of the application of these topics. Next slide. The actual NOFO include a list of non-responsive applications and the topics, but here I listed 3 specific research topics that I'd like to bring up to your attention. The first is the research narrowly focused on pathology of specific viral infection and specific disease state that are not part of a broader application centered on defining and characterizing the human virome. This will be non-responsive applications and won't be reviewed. And also the research focused on only the interactions between viruses and microbial hosts independent of a human host. This will be for those applications on the bacteriophage specifically. And lastly, the particular interest on the translational and product research will be considered as a non-responsive applications. Next slide. So these are the few keys for this RFA. The application submission due date is November 15 of this year, and the scientific review will be performed in March of 2024, followed by advisory council in May, and the earliest start date for those awarded projects will be July of 2024. Like the RFA, no late application will be accepted for this funding opportunity. Next. Those are the key budget-related information. The NIH Common Fund intend to commit about 12 million in FY 2024, which will allow us to award about 10 projects. In terms of the award budget, the direct costs are limited to a maximum of 750,000 in each year during the duration of 5 years of the project, which also should include set aside a minimum of 50,000 in direct costs each year for years 3, 4, and 5 for the collaborative pilot project. Next slide.
All right, thank you. Manuel?
Hi, good morning, everyone. My name is Emmanuel Mongodin, and so I'm the scientific contact for the Consortium Organization and Data Collaboration Center, or CODCC, for the HVP program. So this will be funded through U24 clinical trial not allowed grant mechanism, and the RFA number is RFA RM 23016. The key dates for the CODCC, I think the timeline is somewhat similar to the timeline for the other program, except the applications for the CODCC are due on November 29, with a letter of intent due on, the application due date is November 29, letter of intent October 29, and the scientific merit review, advisory council review, and earlier start date will be similar to the other program. And I invite you to read all the details in the NOFO. And for any specific question, please don't hesitate to send an email to the human virome address listed here, or to contact the scientific program listed on each specific NOFO. Next slide. So the CODCC will have 2 main responsibilities. The first one will be to oversee the organization and the management of the HVP consortium. And the second responsibility will be to coordinate the data acquisition, analysis, and integration of the HVP outputs from the various consortium components. So this would include the creation and the maintenance of the HVP data portal that will also be used to house the human virome reference dataset, which will be a non-redundant catalog of human viral sequences that will incorporate potentially the data generated by the HVP consortium plus any other human virome data that might already be out there. The CODCC will work, we anticipate that the CODCC will work with a variety of data types and tools that will be generated by projects from the current HVP, so under the companion NOFOs that we talked about, but also by future HVP funding opportunities. The CODCC will also be tasked to provide training, documentation, and technical support for end users within the consortium. One key aspect of the CODCC will also to promote collaboration and communication among the HVP investigators and coordinate the standardization of protocols and data sets that will be used and generated by the HVP. And finally, the CODCC will have the responsibility to coordinate outreach activities. Next slide, please. So the overall purpose of the CODCC will be to serve as an organizational and cooperation hub for the HVP consortium. So meaning that that hub, the CODCC, will be central to collect, store, curate, disseminate pretty much everything that will be generated and created as part of the HVP, whether it be data, metadata, analysis, visualization tools, computational models, and all data that will be generated or put together across the consortium. And all of that needs to be collected and stored into a searchable HVP data portal. The CODCC will ensure the utility of the database, so internally at first, but we expect that a lot of these things that will be collected and stored in the HVP data portal will eventually be publicly available externally. And as I mentioned earlier, a key point here is the CODCC will facilitate collaboration through the HVP consortium engagement, not only among the HVP consortium, but also with the broad research community. Next slide. So in terms of specific activities that we wanted to highlight from an administration and management point of view, we expect that the CODCC will identify one or several administrators to dedicate substantial effort to administrative core activities and collaborate closely with the CODCC PDPIs, NIH staff, and the rest of the HVP PDPIs. The CODCC will be tasked to establish a consortium working group or working groups as needed to develop common protocols and standards on specimen collection, including, for example, things like consent language. Also protocols and standards for processing, storage, and analysis of HVP samples and data sets. The CODCC will develop a virtual repository of biospecimens, tools, and regions. And we expect that the CODCC will collaborate with NIH staff to organize HVP scientific meetings, which will include the HVP kickoff meeting that will take place 3 months roughly after awards are made, and subsequent annual 2 to 3 day in-person HVP meetings. Next slide. Other administration and management tasks would include arranging and supporting travel costs of approximately 6 NIH-appointed external program consultants to the annual consortium-wide grantees meeting, organize and coordinate HVP governing and advisory bodies, which will include the HVP steering committee and subcommittees as needed. Also, HVP working groups to support some of the HVP activities, and also external scientific panels. The CODCC will facilitate other HMP activities, such as small topical workshops, collaborations with outside group, implementation of some of the HVP developed policies, and things like, for example, consortium opinion and perspective pieces, or things that could be, for example, article providing updates on the HVP activities throughout the direction of the Human Virome Project. So in a nutshell, we expect the CODCC to be like, if you want, the perfect virome, meaning that we would like the CODCC to evolve, adapt, and improve during the project period, and doing that all in response to the needs of the HVP community. Next slide, please. So in terms of data management, we wanted to make clear that the HVP investigators will be responsible for submitting their own data, so meaning that the data they generate to publicly accessible repositories, for example, NCBI. But in that process, the CODCC will have a role to help facilitate, curate, disseminate data, metadata analysis, and visualization tools, computational models, completed viral sequences, etc., that are generated by the HVP consortium in a manner that ensures interoperability with data from other Virome efforts. We expect that the HVP investigators, in addition to submitting their own data with the help of the CODCC, will also submit their data to the CODCC so that that data can be assembled and visualized into a searchable Human Virome reference data set that will make publicly available. And the CODCC will also ensure that common data elements are used throughout the HVP, that standards are either adopted or put in place. And the CODCC would also ensure that specific data requirements, for example, are in place and used by the HVP. Next slide. Among other selected activities of the CODCC will be ensuring open and controlled HVP data access. So the CODCC will establish appropriate data sharing policies and data access processes for the HVP consortium to review and approve before these are implemented. The CODCC will be responsible for the creation of a robust system that can distinguish between and segregate control access versus open access data. And in terms of data analysis and integration, the CODCC will ensure that the knowledge base is flexible and can be scaled up to visualize, manage, and integrate multiple types of data. So obviously, at this point, we do not know what specific data set the CODCC will be tasked to handle, but we expect that data sets that will contain genomics, transcriptomics, molecular and cellular imaging, structural data, epidemiological and clinical data will be generated by the various projects, and that the CODCC should be able to handle a wide variety of data. And finally, the CODCC will also be tasked to facilitate data use not only by the HVP consortium, but also by the broader scientific community. Next slide. So this is a reminder that a data management and sharing plan is required. And so this is pretty much for every NIH grant effective for due date on or after January 25 of this year. So there is a new data management and sharing plan that has to be attached with Form H in the application form package. So the CODCC application is no exception. But we, despite that this DSMP is required, we also would like application to include in the main application all sections as outlined under the PHS 398 research plan that describe basically the overall CODCC vision and management, section 2 administration and management, so including consortium coordination and outreach, and under section 3 a data analysis and integration section that address aspects such as data standards, storage, and dissemination. Next slide, please. And so I believe this is my last slide. So these are the members who participated in the working group developing the CODCC NOFO. So myself at NHLBI and also Ximen Zhao, but so we also have members from various ICs. And again, if you have any question, feel free to send us an email via the HumanVirom at od.nih.gov mailing address, or contact any of us specifically. Thank you.
All right, thank you. And I'd just like to thank our working group as well. We have a large number of NIH staff who have helped put this program together, so I'd like to thank them for all of their work as well. All right, so now we are moving on to the next portion of this webinar, which is Q&A. So we ask that everyone please use the Q&A box to submit your questions, and use the chat for any technical issues you might be having. Once again, we're not going to be discussing anything that's very specific to you scientifically or situationally specific. We'd like to keep the questions of broader relevance to the wider community, but please email us directly for those questions you do have. If you have those additional questions, you can reach us at humanvirom@od.nih.gov. And as a reminder, this webinar is recording, and we will be posting the recording and the slides on our website at commonfund.nih.gov/humanvirom. And also on our website, you'll be able to find FAQs that can also provide additional information. So for this portion, I'm going to open it up. So please go ahead and submit your questions. For the panelists, you can view your Q&A box on the bottom right of your screen. There's the 3 ellipses. One of those options there is the Q&A box, so you can open that so you can actually see the submissions. And Sonika, do you want to go ahead and let's get some of the first questions going?
Yes, so this question is directed to Roberto or Stacy for the Virome Characterization Center. Is there a vision for how much of the U54 funds should go to biospecimen analysis versus collecting the cohort samples?
I don't know if Roberto, you want to answer, or would you like me to answer?
You can answer, Stacey, it's fine.
Yeah, there is not a specified amount. It will depend on cohorts, the needs of the cohort, whether or not additional samples need to be collected, how many samples will need to be collected. You know, a lot of existing cohort samples may or may not be useful for characterizing the virome. So it's pretty open-ended. I think we would ask that you use your best judgment in terms of what you think you may need in terms of collection from the cohorts that you put forward. And if anybody else has any additional feedback for that question, please feel free to step in.
I think we receive another similar question regarding number of specimens and the statistical considerations for power calculations. And the idea here is that the characterization of the virome is the phase 1 of the program, and in subsequent in a subsequent phase, there will be more characterization of the functional aspects of the virome. So having that in mind, then the question is how many specimens would hypothetically be needed for future studies, considering that we'll be moving towards functional characterization. So that's another aspect that investigators have to consider.
Thank you, Roberto and Stacy. This next question, I believe, can be addressed to all of the panelists. Dear officers, just wondering whether epigenetic modifications will be considered as annotation of human virome.
That's kind of a difficult question to answer. I guess it would depend on what you mean by the epigenetic modification. If you're thinking of changes in the, modifications to the viral genome, that, the genome itself will probably remain the same. You just have additions that may impact protein translation of proteins and things like that. So I'm not sure. We haven't really discussed that, and that may be something that will be discussed during the consortium as a whole, what we would like to see in the database in terms of annotation. So I don't know if Emmanuel might have some additional feedback on that.
Yeah, I agree with you. It's probably difficult to answer without looking at kind of like, the broader characterization that, a specific project would have. But I would have to say that if there is a project that has among the data set that it is expected to generate epigenetic modification, then yes, I would say that, we would expect the HVP to include that into the set of data that is captured by the overall program, and also the data hosted by the CODCC. So I'm not sure that answers the question, but yeah, that's probably my answer.
Thank you, Emmanuel and Stacy. The next question is for Emmanuel. Our assumption for the CODCC has been that the virtual biorepository is essentially a loose catalog of specimens that are individually banked internally to the various projects. That means it's neither comprehensive nor a strict LIMS, as would be the case for a real biorepository, if that's the program expectation.
Yes, probably, although I think that, we're starting to dig into kind of like the details of how this biorepository would be implemented. But the way we envision that biorepository would be, yes, as a catalog of all the biospecimens that have been characterized by the CODCC across all the HVP projects, and provide a comprehensive overview of all these specimens, what has been done to them, and potentially, who has them or who might have some leftovers. So I think the type of questions that could be answered, and that's just an example, would be, something that would be searchable. And if someone at some point would like to see the type of samples that have been characterized containing, virus X, you run that query, through the CODCC portal, and that gives you, all the samples that, for example, that virus was encountered, and then you can make a decision, whether or not you want to retrieve the sequences, or if you might be interested to see, some of the samples that might be left, for example, that have been characterized, etc. So the idea is to make it easy for people, if they have an idea, to find the right person to collaborate with, and have some cross-project collaboration this way. So I don't know if we would call that, a loose catalog or something more specific, but that's the idea. The idea behind it, to promote collaboration and making sure that if someone has a project or a specific question that might be answered by a set of samples that have been processed and characterized with the CODCC, that that information can be easily retrieved.
Thank you, Emmanuel. The next question is a recurrent question, but it's specifically focused on RFA RM 23-0117. Heysuk, I believe this is your RFA. Can individual PIs apply, or must it be multi-PI?
You could be multi-PI, and also it could be single PI. We accept both.
Thank you. This question, the next question, is for all panelists. With the goal of establishing harmonized protocols across the consortium, if this includes sample collection, handling, storage methods, then this can only apply to prospective cohorts and new samples. Is this a correct interpretation, that the focus should be on new sample collection?
Take myself off of mute. So we would like to see samples from existing cohorts where it's possible. We understand that the samples were not collected for the purpose of the Human Virome Program, and so may or may not be useful for this purpose. Keeping that in mind, we put together the program with the expectation, or we feel like, the possibility that new samples will need to be collected for the existing from the existing cohorts is likely. And so we expect that any applications that come in response to the U54s where you're collecting samples will include new samples as well. So I guess the question is prospective sample collection from existing cohorts is what we had in mind.
Thank you, Stacy. Heysuk, this next question is for you. Would identification and characterization of viral non-coding RNAs be considered a fit for RFA RM 23-0117?
Yes, it is. They are included, yeah, as a human virome.
Thank you. How do we describe inter-consortium collaborations if the consortium members are not known?
But this is in regard to the U54. We understand that we don't know who the awardees will be, but it is suspected that all the characterization centers work synergistically. So once they awarded and the consortium is formed, then there will be an initial phase when a steering committee will be formed, and then they will establish how collaborations and how the connectivity will be among the different characterization centers.
Thank you, Roberto. When stating diverse population, are you looking for one of the diverse population or a center that collects samples from multiple diverse groups? Roberto, I believe this one is for you as well.
Yes, can you repeat the question, please?
When stating diverse population, are you looking for one of the diverse population or a center that collects samples from multiple diverse groups?
Well, ideally, each application would include diverse populations, and we define diversity in terms of gender, race, socioeconomic status, geography, and other parameters. So we strive for all the characterization centers at least to cover a wide range of diversity populations and parameters. So, so ideally, each one of the centers will come up with some diversity parameters, so we can cover as much as possible.
Okay, thank you. And the next two questions are for you, Roberto or Stacy. Do all U54 samples need to be longitudinal, or can some be cross-sectional across different age groups?
I think it will depend on the nature of the biospecimen collected. Some of them will be easier to collect longitudinally, and some may be more invasive. And we will not expect that an invasive collection of biospecimen will be ethically possible in a healthy population. So cross-sectional in that case will be acceptable.
Great, thank you. And then the next question states, is there a specific expectation on which kind of data HVCCs will generate, like mass spec, genome or exome sequencing, sickle cell RNA sequencing, ATAC sequencing, etc.? Stacy, we can't hear you. You're still on mute.
Sorry. Yeah, there is no expectation on the types of technologies that will be used to characterize the virome. I think it will depend on the types of samples, the tissues where the samples are being collected from. You know, we would hope that each center will use the technologies that are available to be able to characterize the virome in its entirety. So as detailed as we can get from technology that's currently in use to interrogate the virome or viruses. So I think we're open to all sources. And in addition, I think in collaboration with the tools and technologies and methods groups, we can refine and build on existing tools in order to be able to get at the question of what the virome looks like and its dynamics.
Thank you, Stacy. The next question is for Heisuk. The NOFO says that non-responsive applications include those that look at specific disease states. Would proposing to look at virome or bacterial or fungal host interactions across various underlying conditions be out of scope or considered non-responsive?
That's very complex question. So we allow the use the specific disease state as a control. But if that the study is solely designed and address the pathophysiology of the disease by the specific viruses are not responsive. I think it depends on it really depends on the what types of the study you propose. So for those who has a similar question, just please reach out to me. We can discuss offline.
Thank you, Heesuk. This question is also addressed to you or others if applicable. Can you provide an example of a collaborative pilot project? Will this be limited to healthy states as well? This sounds like a complex question as well that probably could be answered via email. Heesuk, would you agree with that statement?
Oh yes, I think it's really depending on the types of the proposed study. And the collaborative, as we discussed here, it could be between the project in different initiatives, and then within the same initiative. So that answer will be very different from the types of the study. So again, if you have any questions specific, then reach out to one of us.
Okay, thank you. The next question is addressed to all panelists. What is the expected time commitment for key personnel to work on working groups? Are all PIs expected to commit to working groups or only those within their expertise?
I might take that one. I don't think we have. So the working groups will be formed on a need basis, and it will probably depend on the type of project that are funded. So this being said, we do not know yet how many working groups will be needed and which ones there will be. So I think that this is a question that will probably be better answered after all the awards are made, and right now we just don't know. So the key here is that we expect all members of the consortium to collaborate one way or another with each other, even if that, through working groups, through, research projects and so forth. But in terms of who's going to be expected to participate in working groups, I think that this is something that will be established most likely at the beginning when all the awards are made and we have a better idea about the type of projects that are funded.
Thank you, Emmanuel. For the U54, is the amount 20 million per year in fiscal year 2024 as per the answer here to one question, or 22.8 million as per the RFA?
Is what the RFA states, but it's the amount per fiscal year. So that will be in fiscal year 2024 and also in subsequent fiscal years, 2025, 26, 27, 28.
Thank you, Roberto. Again, for the U54, since the pilot projects are expected to be collaborative with with other centers, are potential pilot projects needed to be proposed in the applications as examples?
Yes, we expect pilot projects to be included in the application and also indicate how this pilot project will be collaborative in nature with other VCCs.
So the pilot projects, I guess will develop as the consortium moves along. So you won't have specifics about what those pilot projects are. The expectation is those collaborative projects will occur in years 3, 4, and 5 of the over the course of the project. So at that time is when sort of the details will be worked out. but it would be expected that there's some thought that's given to how those projects will be, the planning of those projects and how they will take place.
Thank you, Stacy. I'm scrolling through the Q&A box right now to identify other questions that were not answered. Can you clarify how we need to set aside $50K for year 3, 4, and 5 for pilot projects when we do not know who else will be in the consortium?
I think
Yes.
this
Yeah.
question was answered by what Stacy mentioned before. I don't know, Stacy, if you want to add anything else.
And I think you just have to keep in mind as you're developing your budget that in years 3, 4, and 5, the expectation is that you set aside $250,000 for each of those years in order to be able to support pilot projects up to $50,000 per year for up to 2 years. So I think it's as you develop your budget, that is a requirement. So just think about that when you're putting your application together.
Thank you, Stacy. The next question, can foreign PIs serve in a multi-PI application or core lead role?
So I don't know if you want to answer that or.
Yeah, I think I don't know if it was this question was before, but I think we're accepting multi-PI applications. So that is acceptable.
Yeah, and a foreign investigator, there's no restriction on whether or not they can lead a core or be involved in a core. I think with all foreign components, you have to provide a good rationale and justification for that foreign investigator being, leading that core, but there's no restrictions on foreign participation in the cores.
Yeah, just to add on to that, there's been a couple of questions about the foreign component and what is the definition of component versus institution. And I think as Stacy mentioned, an institution or PI can be part of core, but it has to be, the rationale has to be really well justified in why this foreign component is used, what are the unique characteristics of this foreign component, institutions, resources, etc., that may bring into the strength or strengthen the characterization center, and how is that relevant to the U.S. cohort in terms of using biospecimens from foreign institutions too.
Thank you, Roberto. The next question was sent through the HVP mailbox and is addressed to Amanda. This NOFO is focused on 2 areas. 1, development of innovative and novel tools, models, and methods to overcome the major challenges in identifying and characterizing human viruses. And 2, development of computational biology and bioinformatics tools to enhance the analysis and visualization of the human virome. For area 1, is it focused on developing tools, models, and methods for human viruses? Does this mean that developing tools and methodology for bacteriophages in the human virome would be non-compliant?
Thank you. I think this was partially answered in the question and answer area already by Haesuk, but essentially the tools to be focused on the human virome, that doesn't exclude bacteriophages if you were happening to be looking at uncharacterized regions of the viral genome, for instance, bacteriophage might be included. But if you're specifically developing tools for specific bacteriophages that are in the interest of what a bacteriophage does and killing bacteria, then that would not be compliant under the human virome. So it really should be focused on the human virome, but bacteriophages aren't completely excluded. If anyone wants to add anything, because I know that Haysuk also has questions on this area, please feel free.
Well, I think you addressed them all. For the bacteriophage in the functional study, I'd like to echo that the study has to be address the human host as well as the bacteriophage interaction with the bacteria host. That's the key things.
Thank you both. The next question was sent through the mailbox, but I believe it might have been sent to the Q&A box as well. The primary objective of the LC core is to protect participants. The RFA states the primary objective of the LC core is to protect participants, investigators, and NIH staff in matters of privacy, safety, and legality by providing guidance on LC and policy issues. Generally, LC focuses on participants. What privacy, safety, and legality issues are anticipated for investigators and NIH staff?
I guess the primary role of the LC will be on the participants, but there may be issues that come up that may impact the PIs and NIH in terms of maybe not privacy, but in terms of overall policy. So if, for example, if there's some issue that comes up with the virome in terms of its use in things that we didn't anticipate, such as healthcare premiums, or health insurance, or life insurance. I think we would hope that there would be some way we can sort of blunt that the impact of those types of things moving forward. And the folks with sort of an LC background may be able to help us navigate through some of those issues. So it's not about protecting our privacy or our safety necessarily, but more on the policy side, if that makes sense.
Thank you, Stacy. Is collection of new samples required for U54 applications, or can they be composed of existing samples only?
Could be existing samples or new collection of samples. It will depend on what's required in order to be able to assess the components of the virome in those samples. So you may have high quality samples that can be used to interrogate the virome. In other cases, new collection may be necessary in order to get at the answers that we are looking for. So it could be a combination of both. And a lot of that will be worked out post-award.
Is there any particular expectation for the number of individuals or samples collected and analyzed by HVCC? Should it be approximately 100, approximately 1,000, approximately 10,000?
I think we answered this question before. We're not expecting a specific number of samples. That will be determined by the investigators and what they're proposing and how they rationalize their the study design.
Thank you. The next question is for Haysuk. Would evaluating the impact of therapeutic bacteriophages on resistant bacteriophages that already exist in the human host and other microbiota and the human host be non-responsive? Or is this a complex question best suited for email?
Oh, yes. I think I can be more specific to this question and by the email. So send me the specific question.
Do we have to have some legal or community personnel in LC Core?
The required composition of your LC core is up to the investigators and what you would like to put forward for the VIROM program in terms of expertise. It's probably a good idea to have someone with some background in both of those areas, but we will leave that up to the applicants to decide.
Thank you, Stacy. The next question is for Emmanuel. Do you have any guidance on the expected volume of data to be generated by the HVP sites?
A short answer is no. I think it will depend on the type of data that the HVP consortium as a whole will generate. So that's why we're expecting the CODCC to be able to scale up and be adaptable. If we compare to what happened, for example, with the Human Microbiome Initiative, I believe most of the data was sequencing data for the most part. I would expect that for the human virome, the data that will be generated will be way more diverse. So probably we will get, a fair chunk of sequencing data, but I could see, some imaging data, for example, also being generated. So, in terms of volume of data, no, we do not know yet, and that's why one of the emphasis that we try to convey in the CODCC presentation is the scalability and the adaptability of the CODCC to, handle pretty much anything that potentially the HVP projects can throw at the CODCC.
Thank you, Emmanuel. The next question is assigned to you as well. Under Section 3, Data Storage and Access, the NOFO states that, however, while the HVP CODCC will host and share all HVP data within the consortium, the HVP CODCC will only distribute open access data to the public. But the section on responsibilities of the CODCC states that because the HVP CODCC will only distribute open access data to the public and will rely on other NIH repositories for distribution of controlled access data, the HVP CODCC will not become an NIH trusted partner. Are these two statements consistent with each other? Will the CODCC be responsible for storing and distributing controlled access data to other HVP members and NIH designated individuals?
Yeah, that's a long and complicated question. So by that, we meant that, for example, it is possible that, or I would say highly likely that HVP investigators will have to deposit data, for example, in dbGaP that will need to be controlled access. So in cases like this one, we will not expect the CODCC to host, obviously, the dbGaP data. However, the CODCC will be able to say or to have a link saying that that data is hosted at dbGaP, maybe with some accession number, where a PI can just follow that link and then request that data, if possible, through dbGaP. So we don't anticipate the CODCC to store everything and anything, But I think that when there is some data that requires a controlled access, then what we will want is the CODCC to point to where that data is hosted, and also maybe have some information about what is required for PI that might be interested to potentially access that data.
Thank you, Emmanuel. The next question is for Amanda. Is setting aside 50K in year 3 also required for pilot projects?
I answered this in the Q&A, but
Okay.
for the tools RFA, no. So it's applicants must budget funds in their final year for cross-validation of their work product, but the amount was not predetermined because it may range greatly since it's for cross-validation.
Thank you, Amanda. The next question is for Heisuk. Are you seeking hypothesis-driven or just descriptive proposals? Relatedly, much work in the human microbiome is only correlative. How much causality and or directionality between virome and host are you seeking?
So I guess the answer is really depending on what types of the interactions, the virome interaction that you are asking. So if there's enough data to go into the causality related question, you can do the hypothesis driven application, but it's a newly and never done any study, or like to do the identification of the specific viral interaction with the host, then it could be descriptive. So it's really depending on the types of the study you propose.
Thank you, Heisuk. This question is a general question. My question is about the expectations of key personnel to participate in consortium activities, including meetings and working group. I believe this question might have already been answered. Do key personnel have a specific requirement for participation, or is it voluntary based on their specific expertise? And again, the question was answered previously by Emmanuel. It is currently difficult to know because the consortium hasn't been set up, and we will know once the awards will be made. Can you comment on the composition of the review panel?
I can answer that. So each RFA will have a special emphasis panel set up for review, and the composition of those panels will be made up depending on what type of applications come in under each RFA to bring in the appropriate expertise.
We actually have Dr. Singh here, who is
Perfect.
from the Center for Scientific Review. Would you like to answer that, Dr. Singh?
Can you all hear me?
Yeah.
Yeah, actually, I think someone already answered that. I mean, there are review criteria there and listed for each NOFO, and the panel will be a reflection of the expertise needed for the application submitted for each NOFO. Thanks.
Thank you, Dr. Singh. I do not see any other questions in the Q&A box. And I believe we've covered most questions that came through the mailbox. Some of them will require direct conversations or discussions between the PO and the author of the question. I think we've reached the end of the Q&A session. Back to you, Becky.
All right, thank you all. So as Sonika said, if you have existing questions or you have follow-up questions, please email us at humanvirome@od.nih.gov, and we'd be happy to work with you on getting those answered. As a reminder, yes, a recording of this will be, and the slides will be posted on our website at commonfund.nih.gov/humanvirome. So give us a couple of days to get that posted, but you will find it there. That's all I have, but feel free to reach out to the scientific contacts listed at the very bottom of the funding opportunity as well. We will also forward them any emails you send to the Human Virome mailbox, but if you want direct contact, you can also reach out to them. All right, well, thank you everyone. Thank you to our panelists for joining us today. We appreciate it, and we wish everyone the best of luck on writing their grants.